All right, the last topic that we want to cover in this chapter is going to be gene therapy. Gene therapy involves procedures to give healthy patients genes to make up faulty genes. It's also going to include the use of genes to treat genetic disorders and various human illnesses. Now, there are two methods of gene therapy that we're going to discuss. There are ex vivo, outside the body, or in vivo methods of gene therapy. Ex vivo gene therapy is the process by which cells are going to be removed from the body, treated, and then returned to the body. So the treatment occurs outside the body, often used to treat children with severe combined immunodeficiency disorder. In this case, these individuals lack mature immune cells and are often prone to infection. So the bone marrow cells are going to be removed from the body, infected with a virus that's carrying the normal gene for ADA, and then it's going to be placed back inside the body once it has been treated. So that's how ex vivo gene therapy works. We take the organ out of the body, treat it, and then put it back in. In vivo gene therapy is going to be a method where we deliver the altered gene directly into the body itself, and it's going to be used to treat whatever diseases. Now, there have been some advancements in the treatment of cystic fibrosis by using a nasal spray. So if you remember, cystic fibrosis is a genetic condition that results in a deficient chloride channel protein. It's going to be responsible for moving chloride ions across the cell membrane inside the lung epithelial cells. With the flow of the chloride ions, we also get a movement of water molecules moving down the concentration gradient to follow those chloride ions. So it's because we have that faulty protein channel, we can treat it by using a nasal spray or a respiratory spray to spray liposomes or lipid globules that contain those lentiviral vectors. These are going to contain the gene necessary to produce the correct protein ion channel. So current research involves the insertion of a p53 tumor suppressor gene into cancer cells to make tumors. Oh, I'm sorry, this is a separate thing from cystic fibrosis. Current research involves the insertion of the p53 tumor suppressor gene into cancer cells to make tumors more responsive to chemotherapy. There are a lot of different ways that gene therapy is being used throughout the body. So just spend a little bit of time with this diagram. It's going to go through gene transfer by injection. So gene therapy has been used to treat Huntington's disease, Alzheimer's, Parkinson's, brain tumors. In the skin, we see gene transfer by modified blood cells to treat skin cancer. In the lungs, we can use gene transfer by aerosol spray to treat cystic fibrosis or hereditary emphysema. In liver, we can use gene transfer by modified implants to treat familial hypercholesterolemia. In blood, we can treat bone marrow to treat sickle cell disease. Endothelium, we can use treat the blood vessel lining. We can use that to treat hemophilia, diabetes mellitus, so on and so forth. I'm not going to read through all of them, but there are a lot of different genetic disorders that can be treated by gene therapies, and depending on what the disorder is, they're all going to have different injection methods, just depending on how we can or how we need to get that gene to the affected organ as quickly and as efficiently as possible. All right, in this chapter, we talked about gene cloning or DNA cloning and gene therapy. If you have any questions, comments, or concerns, or anything you'd like to discuss in our discussion groups, please feel free to shoot me an email, and I will get back to you within a day.